So we know that humans have a genome size of around 3.2 billion base pairs and about 20,000 genes. And what's interesting about that is that this is actually a lot fewer genes than scientists originally expected the human genome to have. And so only 1% of the genome, or the full set of DNA that humans have, is exons. And remember that exons are the part of genes that actually become protein. So only 1% of all of our DNA sequence is ultimately destined to become translated into protein. 24% of our genome is made up of introns, which are the parts of a gene that actually get spliced out before it becomes an mRNA or becomes a protein. And so that means that if you take this 1% of exons and 24% of introns, you only really see 25% of our genome being made up of protein coding genes, or genes that actually become a protein. And that's only 1/4 of the genome, right? And so what is all of the other stuff? Well, we used to, sometimes we now kind of mistakenly referred to all of the 75% of the other DNA that was not a protein coding gene as junk DNA. But junk is really kind of implying that there's absolutely no function to any of that other DNA. And the more that we understand genomes and more that we understand DNA sequence, we realize that there's really not junk DNA. There is repetitive DNA. Over 50% of our genome as humans is repetitive, which means those sequences are seen over and over again. But it's not to say that those sequences don't have any function or any use at all. It's possible that we haven't determined the use of that quote unquote junk yet. And so if we look at this figure here, it shows the breakdown of what kind of structures are inside of the genome. We can see exons and introns making up about 25% of this genome. And then we can see the rest is all different types of repetitive DNA. So what we also have come to understand from studying the human genome is that our genes aren't distributed evenly on chromosomes. And so this is just an image that shows each individual chromosome from chromosome 1 all the way down to the sex chromosomes X and Y here on the right. And then it shows the number of genes on each one of these chromosomes. So chromosome 1 has the most genes with over 2,000. Chromosome 18 has very few genes. Chromosome 13 has very few genes. Chromosome 21 has very few genes. And the X chromosome has, or the Y chromosome, has the absolute least number of genes. And one thing that I like to point out that's kind of interesting is that there are several genetic disorders where there's a complete lack or extra copy of a chromosome. We talked about these, right? There are monosomies where one chromosome is completely missing, one copy. And there are trisomies where there's a completely additional copy of a chromosome. 3 of the most common trisomies that are observed are on chromosomes 13, 18, and 21. Chromosome 21 trisomy would be Down syndrome. Right? And so what's interesting is it seems like these chromosomes that have quite small numbers of genes, your body can actually tolerate an extra copy of the chromosomes. And the zygotes that have these trisomies for chromosome 21 or 18 or 13 can actually remain viable. Whereas a trisomy of chromosome 1, we have never seen a patient who has that. So we know that there are less genes than we thought, about 20,000 genes in the human genome. We know that those genes aren't like evenly distributed across the chromosomes. There's some chromosomes that have more genes and some that have less. And what scientists really wanted to know about genes is how many of them are absolutely, absolutely essential to the function and viability of the organism. Because yes, you have 20,000 genes as a human, but do you need every single one of those 20,000 genes? Or is it possible for you to live with only 10,000 or 4,000 or 1,000 or 100, right? And so we've been studying this idea of essential genes and like the minimal number of genes that are needed for an organism to survive. And we used to use lower eukaryotic organisms like yeast or like C. elegans, the worms that I study, to determine how essential genes are. And so this image here is look as an image of basically worm genes and how essential or non-essential that they are. And what they did was they took every single worm gene and they deleted them one by one, and then they looked to see what happened to the worm. And so they made their way methodically through the worm's 20,000 genes, and they slowly deleted them one by one. And what they noticed is that in 90% of those worm genes that they deleted, they were not able to see any phenotypic or any visible differences at all. No detectable effect on how the worm looks, behaves, moves, any of that stuff. Which means that 90% of their genes were not essential. The other about 10% did show some effects when they were deleted. Some of them, 7%, caused the worm to die when they were deleted, and some of them caused other phenotypic effects. And so that's really interesting because it would suggest that even though your genome may have this large number of genes, there are only a certain set of them, in this case about 10% for worms, that are essential. And so based on studying worms and other organisms, we estimate that the number of essential genes in humans is not the 20,000 that we have, but rather something more like 4,000 to 8,000. Things that if we deleted them, we would die or have a very visible phenotype. Right? And so you can actually see a breakdown of how they determined this number, this 4,000 to 8,000 here in this image as well, by looking at knockout mice, human populations, and human cell lines. And that's how they got to these numbers of somewhere around 4,000 to 8,000 essential genes. And so we also wanted to know how many genes are unique to particular organisms or how many are similar. So we all know that we come from a common ancestor, right? And so we think that there's probably some genes that are going to be shared or at least very similar between our common ancestor and us, between flies and us, between C. elegans or worms and us as humans. And so we are totally right in assuming that there are some genes that make similar proteins and even have a similar sequence in different organisms. And we refer to these kind of genes that are similar between two different organisms as orthologs. They are orthologous to each other. They are similar in a fly and a worm. They're similar in a fly and a bacteria. They're similar in a worm and a human. They're orthologous, right? And so what you can see here is the total number of genes in each one of these different organisms, from bacteria all the way up to humans. And you can see that that number of genes increases as the organism gets more complex. But what you'll notice is that the number of distinct or unique genes does not completely match up with the number of total genes. And so in a bacterium, the number of unique genes is almost equal to its total genes, which means that every single one is unique. But then when we look at yeast, we can see that only about 3/4 of those genes are going to be unique to yeast. Other ones are going to be orthologous to something else. In the fly, you can see like almost all of them are unique. But as we get up to humans, what we'll notice is that we actually have a smaller number of unique genes than we might think. It's not really up to 90%. It's almost at like 50% uniqueness, which means that we have a lot of genes that are orthologous to these other organisms, which is great, right? Because that means that we are able to use those organisms as genetic models to study our illnesses and diseases. And one other thing that this graph shows is trends that as genome size increases, the proportion of unique genes or new stuff decreases, and the proportion of related genes increases. So as the genome size gets bigger, basically there are less and less unique genes as compared to genes that are related to genes in another organism. And you can see that laid out in this table right here. This is a bacterium, H. influenzae, yeast, fruit fly, worm, and then Arabidopsis, which is a plant genetic model organism. And you can see that in the smaller genomed organism, bacteria, there's 89% unique genes. And about 11% of related genes. But as you increase the genome size up to an organism with a large genome like Arabidopsis, you'll see that now the number of unique genes decreases, and those number of related genes or orthologs increases. Meaning that Arabidopsis doesn't have as many unique genes as Haemophilus influenzae. It relied on evolution to create some other genes that are orthologous. Why reinvent the wheel? Let's just use a gene that's similar to something that came before. And so one other thing that I just want to touch on about the size of genomes and complexity has to do with this thing called a C-value paradox. And so what a C-value is the total amount of DNA in the genome. So the total number of base pairs or nucleotides is the C-value. It's an indicator of genome size. And so the C-value increases as organisms get more complex. And what you notice if you look here, this is organisms in increasing complexity as you move up, and you can actually see just the number of nucleotides here on the bottom. So you can see that there's a general trend of there being more nucleotides as organisms get more complex. But that's not completely true. There's a general trend, but there are some situations where that relationship does not hold. And there's a lack of relationship between how big a genome is and how complicated that organism is. Right? And so we can see that relatively well here, looking at the flowering plants. Flowering plants are less complex than mammals. That's just true. They are not as complicated. They do not have as many physiological processes as mammals, including humans. But what we see is that they can have a humongous genome, all the way up to 10 to the 11th nucleotides. And that number of nucleotides does not correlate directly with how complex that organism is. So generally, there's a trend where there's more complexity and an increasing genome size, but there are some specific examples where this does not hold true. And in those instances, that's what we call a C-value paradox. There is no relationship between how big the genome is and how complex that organism is. And the way that organisms really add complexity is not necessarily by adding genes, it's by adding functions to those genes. And so we have come to understand that complex morphology doesn't necessarily have to mean more base pairs. It might mean those base pairs are rearranged in a different way to create more genes and allow that complexity to increase. So there is a correlation between how complex an organism is and how many genes that it has, right? So organisms add genes, increase their number of genes to add complexity.